he'd been exhaling, thereby causing an explosion since excess oxygen combined with hydrogen was highly inflammable. Later on, he puts on his EVA helmet with a radioactive suit as he marches toward the water plant. He repeats the previous steps he'd taken while setting up the water plant. However, it doesn't blow off this time. Mark wipes his hands on the water produced on the walls of the tent as the water spreads over the soil. He checks in on everything regularly, and he also adjusts the solar panels outside the hab. He makes some repairs to his EVA helmet, and on Sol 54, he notices a little sprout of the plant inside the tent when he passes by with his plate of food. He then goes over to say hello to the little plant. Back on Mars, Mark cultivates his potatoes after 48 sols, and they grew even better than he'd expected. He carefully uproots them, after which he keeps the plant to further use it. Log entry 79, he's now become the first one to grow healthy potatoes on the Martian soil. Receiving lots of emails, and how his alma mater had sent him some tips for his farm. They'd also told him that he'd colonized Mars since he was the first to plant on the Martian. He calls himself the best botanist, and he also talks about the message he'd received from NASA, asking him for a photograph on his next transmission. Afterward, he begins practicing how he'd pose.
How can we optimize the ingredients? Are these algae useful for the remediation of wastewater? Can we predict the growth of certain algae based on our collected data? These are all questions to which we try to formulate an answer. Microalgae are grown in a fresh or salt growth medium that contains necessary elements such as nitrogen, phosphorus and trace elements including iron, copper and zinc. In our cultivation chamber, the temperature is kept constant and algae are aerated. When sufficient biomass is grown on a laboratory scale, the biomass is transferred to our pilot reactors in the greenhouse. Our pilot installation SynBuild consists of 4 closed photobioreactors that are designed according to a fruit tree model so that the illumination of each tube is optimal. Air and carbon dioxide are blown into the reactor by using a pump, which also creates a wave in the reactor. This wave action ensures that the gas exchange in the reactor is optimal, the algae are well mixed and the deposits on the tubes are limited. Various sensors have been built into the reactor that collect data about the growth conditions of the algae, such as the pH, turbidity, carbon dioxide consumption, etc. The climate in the greenhouse is also closely monitored and controlled. The nutrients are prepared in stock vessels with the help of liquid fertilizer that are common in horticulture. The algae are inoculated in the reactor vessel and can now continue to grow until the desired density is reached. Samples are taken at regular intervals during the cultivation. These samples are examined in the lab to monitor the concentration of nutrients in the medium and to check the culture for the presence of contamination. The dry weight is also monitored through sampling. The algae are harvested using a centrifuge that reduces the concentration of water in the biomass to about 10 to 30%. The algae paste is then further dried using a freeze dryer to obtain algae powder. Radius conducts research on medium circulation in close collaboration with Vito so that water and nutrients can be reused and the algae story becomes even more sustainable. Microalgae do not compete with agricultural land and show a higher productivity than agricultural crops. Many microalgae thrive in a medium with a high salinity such as seawater, which is abundantly present. By using side streams such as heat surpluses and carbon dioxide and the recirculation of the water used,